In the name of Allah, the most merciful and beneficent. Assalamu alaikum class. I'm Ramsha Khanam from Chemistry Department and my ID is 001. Today I'm here to represent my presentation and my presentation topic is X-ray diffraction. This is a diagram of X-ray diffraction. Now I discussed basic outline of X-ray diffraction. First one, introduction to X-ray diffraction. Second, theory of X-ray diffraction. Third, X-ray diffraction techniques. Fourth one, application of X-ray diffraction. Fifth one, advances and future directions. And the last one is conclusion. Now I discussed introduction. Basically, X-ray diffraction is used to obtain structural information about crystalline solids, useful in biochemistry to solve the 3D structures of complex biomolecules, bridge the gaps between physics, chemistry, and biology. X-ray diffraction is important for solid-state physics, biophysics, medical physics, chemistry, and biochemistry. Now I discuss definitions and basic principles of XRD. Definitions. XRD or X-ray diffraction is a technique used to study the structure and composition of materials at the atomic and molecular level. It is based on the principle of X-ray scattering by crystals and it provides valuable information about the arrangement of atoms or molecules in a material. Now basic principles. Diffraction. X-ray diffraction refers to the scattering of X-rays by the atoms or molecules in a crystalline material. When a beam of X-rays interacts with the crystal, the X-rays are diffracted in various diffraction due to constructive interferences between the scattered waves. The resulting diffraction pattern can be measured and analyzed to determine the atomic or molecular structure of material. Diffraction pattern. The diffraction pattern is the collection of spots or peaks observed when X-rays are diffracted by crystal. Each peak corresponds to a specific set of crystal planes and provides information about the spacing and orientation of these planes. Now I define history of X-ray diffraction. Now 1895 X-rays discovered by Roentgen. 1914 first diffraction pattern of a crystal made by Nipping and Vonlea. 1915 theory to determine crystal structure from diffraction pattern developed Bragg. 1953 DNA structure and developed by Waterstone Crick. No diffraction improved by computer technology methods used to determine atomic structure and in medical applications. Now how diffraction works. Wave interacting with the single particles. Incident beam scattered uniformly in all directions. Wave interacting with the solid scattered beams interference constructively in some directions, producing diffracted beams. Random arrangement cause beam to randomly interfere and no distinctive pattern is produced. Crystalline material. Regular pattern of crystalline atoms produces regular diffraction pattern. Diffraction pattern gives information on crystalline structure. This is a diagram of NaCl. How diffraction works schematic. Basically this is a high voltage, this x-ray tube pass to x-ray beam. This is a light screen, so crystal a photographic plate. X-ray diffraction of NaCl. Schematic diagram. Now theory of X-ray diffraction. First one, crystal structure and lattice. Crystal structure and lattice. The theory of X-ray diffraction in crystal structure and lattice can be explained using Bragg's law and the concept of reciprocal lattice. First one, Bragg's law. Bragg's law relates the angles at which X-rays are diffracted to the spacing between the atomic planes in a crystal. It is given by the equation n lambda is equal to 2d sine theta degree, where n is an integer representing the order of the diffraction, first order, second order, etc. Pi is the wavelength of the lambda is the wavelength of the incident X-ray. D is the spacing between the atomic planes in the crystal lattice. Theta is the angle between the incident X-rays and the atomic planes. Bragg's law explains why certain angles of incidence and diffraction produce constructive interference and give rise to distinct diffraction peaks in the X-ray pattern. No reciprocal pattern. Reciprocal lattice. The reciprocal lattice is a mathematical construct that provides a convenient representation of the crystal lattice in reciprocal space. It is a lattice formed by the set of all reciprocal lattice vectors which are normal to the atomic planes in the crystal lattice. The reciprocal lattice is defined by the reciprocal lattice vectors denoted as G. The reciprocal lattice vector are related to the lattice vector are related to the lattice vectors of the crystal denoted as A, B, and C by the following relations. A is equal to 2 pi, B is equal to 2 pi, C is equal to 2 pi. The reciprocal lattice vectors determine the spacing between the diffraction planes in reciprocal space. The length of a reciprocal lattice vector is inversely proportional to the spacing between the corresponding atomic planes in the crystal lattice. By considering the geometry of the crystal lattice and the reciprocal lattice, one can relate the position and intestines of the diffraction peaks in the X-ray pattern to the arrangement of atoms or molecules in the crystals. Now, second, Bragg's law and diffraction pattern. The theory of X-ray diffraction is based on Bragg's law and the analysis of the resulting diffraction pattern. Let's solve into each of these aspects. Bragg's law. Bragg's law explains the relationship between the angles of incidence and diffraction of X-rays and the spacing between the atomic planes in a crystal. It can be expressed as 2d sine theta is equal to n lambda, where n is the order of the diffraction peak integer, lambda is the wavelength of the incident X-ray beam, d is the spacing between crystal lattice planes, theta is the angle of incidence or diffraction of the X-ray beam with respect to the crystal lattice plane. This equation relates the wavelength of the X-rays, the crystal lattice spacing, and the angle at which the X-rays are diffracted. It shows that constructive interference occurs when the path difference between X-rays scattered from adjacent lattice planes is an integer multiplier of the X-ray wavelength. How diffraction works into Bragg's law. This is a diagram of diagram of Bragg's law. N is equal to 2d sine theta. This is the angle X-rays of wavelength. Similar principle to multiply slit experiments. Constructive and destructive interference pattern depends on lattice spacing and wavelength of radiation. By varying wavelength and observing diffraction patterns, information about lattice spacing is obtained. Now, diffraction pattern. Diffraction pattern: when a beam of X-rays is incident on a crystal line space sample, it interacts. It 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 it, inter, it interacts with the atoms in the crystal lattice. According to Bragg's law, the X-ray will be diffracted by the crystal lattice planes, and constructive interference will occur at specific angles after diffraction. The diffraction pattern produced by the interaction of X-rays with the crystal is a series of bright spots or peaks on a detector screen or film. Each peak corresponds to a specific angle of diffraction and is related to the spacing between the crystal lattice planes. The positions and intestines of the diffraction peaks provides valuable information about the crystal structure, such as arrangement of the atoms in the lattice and the distances between them. By analyzing the pattern of diffraction peaks, scientists can determine the crystal structure, crystal symmetry, and lattice parameters of the material under investigation. This is analyzing diffraction pattern. This is a cubic sines. D1 is equal to 1.09 a, D2 1.54 a, wavelength is equal to 0.697 a. This is the intensity second. This is a wavelength. NL is equal to 2d sine theta q. Now, third, scattering and interference of X-rays. Scattering of X-rays. When X-rays interacts with matter, they can be scattered in different directions. The scattering process occurs when X-rays interact with the electrons in the atoms of the material. There are two main types of scattering. One, elastic scattering. In elastic scattering, the energy and wavelength of the scattered X-rays remain the same as the incident X-ray. This type of scattering is also called relief or coherent scattering and is dominant at low scattering angles. It contributes to the background scattering in X-ray diffraction experiments. In elastic scattering. Second, inelastic scattering. The energy and wavelength of the scattered X-rays are changed due to interactions with the atoms or molecules in the material. Inelastic by free electrons and the photoelectric effect, where X-rays are absorbed by atoms and re-emitted at different energies. Interference of X-rays. Interference occurs when two or more waves interact with each other. One, crystallography. When X-rays interacts with the crystalline material, they can undergo constructive or destructive interference due to their interaction with the regularly arranged atoms in the crystal. It is this phenomenon is basis of X-ray crystallography, a powerful technique used to determine the atomic and molecular structure of various substances. B, thin film interference. When X-rays pass through a thin film or multiple structure, interference can occur between the waves reflected from the upper and lower interference of the film. This interference pattern can be used to study the thickness, composition, and properties of the thin film. See, diffraction. X-ray diffraction is another form of interference that arises when X-rays encounter a periodic structure such as crystal lattice or a diffraction grating. The diffracted X-rays produce a characteristic pattern of constructive and destructive interference, which can be analyzed to obtain information about the arrangement of atoms in the material. Both scattering and interference of X-rays have significant application in various fields, including material science, physics, chemistry, biology, and medicine. They provide valuable insight into the structure, composition, and properties of matter at the atomic and molecular scale. This is a now I define X-ray diffraction technique. This is a technique diagram of X-ray, XRD. First one, powder X-ray diffraction. This is a representation of PXRD. Powder X-ray diffraction is a powerful analytical technique used to investigate the crystalline structure of material. It involves you shining X-rays into a powder sample and analyzing the resulting diffraction pattern to determine information about the arrangement of atoms within the material. Here is a brief overview of how powder X-ray diffraction works. First one, sample preparation and handling. Second, instrumentation and experimental setup. Third, data collection and analysis. Fourth one, phase identification and crystal structure determination. First one, now I discussed one by one. Sample preparation and handling. Sample preparation and handling are critical steps in powder X-ray diffraction analysis to ensure accurate and reliable results. Here are some important consideration for sample preparation and handling in PXRD. First one, grinding. The material of interest needs to be ground into a fine powder. The grinding process should be performed carefully to avoid introducing contamination or altering the crystal structure. It is important to use clean grinding equipment to prevent cross contamination between samples. Second, homogeneity. The powder sample should be well mixed to ensure homogeneity. This is important because the diffraction pattern obtained should be representative of the entire sample. Homogeneity can be achieved by through mixing or by grinding the sample for an extended period. Third one, sample quantity. Sufficient sample quantity is necessary to obtain accurate result. Ideally, the sample should fill the sample holder evenly without any gaps. It is recommended to use enough sample to ensure a good signal to noise ratio in the diffraction pattern. 4. Sample holder. The sample is typically placed in a sample holder which can be made of materials such as glass, quartz, or specialized material like silicon. The sample holder should be clean and free from any residual material that could interfere with the diffraction pattern. 5th, sample handling. When handling the sample, it is critical to minimize contact with the bare hands or other sources of contamination. Disposable gloves are often used to prevent fingerprints or oils from being transferred to the sample. It is also important to handle the sample holder carefully to avoid damage or misalignment. 6. Moisture and air sensitivity. Some material may be sensitive to moisture or air. In such cases, it is necessary to handle the sample in a controlled environment such as a dry box or a glove box to prevent exposure to moisture or oxygen. This ensures that the sample remains stable and does not undergo any undesirable changes during analysis. 7th, sample alignments. Paper alignments of the sample is important to obtain accurate diffraction data. The sample should be placed in the X-ray beam such that the incident X-ray are directed perpendicular to the sample surface. Some sample holders have alignment features or reference marks to aid in proper poisoning. 8th one, cleaning. Regular cleaning of the sample holder and the X-ray equipment is necessary to prevent cross contamination between samples. Cleaning methods may vary depending on the specific materials used. Common cleaning techniques include ultrasonic cleaning, rinsing with solvents, or heating in a furnace. By following these guidelines, proper sample preparation and handling can minimize potential sources of errors and ensure reliable and reproducible PXRD results. It is important to consider the specific requirements of the material being analyzed and any potential sensitivities or special considerations during the sample preparation process. This is a diagram of clean. Now, second, instrumentation and experimental setup. First one, instrumentation. The choice of instrumentation and experimental setup depends on the specific nature of the experimental or research project. However, I can provide you with a general overview of commonly used instruments and experimental setups across different scientific disciplines. Second, microscopes. Microscopes are widely used in biology, materials science, and other fields to observe and analyze samples at the microscopic level. Different types of microscopes include optical microscopes, electron microscopes such as scanning electron microscope, and transmission electron microscopes, and atomic force microscope, spectrometers. Second one, spectrometers are used to measure the interaction between matter and electromagnetic radiation, providing information about the composition and structure of materials. Examples include mass spectrometers, infrared spectrometers, UV visible spectrometers, and nuclear magnetic resonance spectrometers. Third, chromatography. Chromatography techniques are used to separate and analyze mixture of substances. Gas chromatography and high performance liquid chromatography are commonly employed methods for separating and identifying compounds in various fields, including chemistry, biochemistry. Third one, data collection and analysis. One, data collection. Once the sample is prepared, it is mounted to on a sample holder and placed in the XRD instruments. X-rays are then directed into the sample at various angles, and the resulting diffraction pattern is recorded. The XRD instruments typically consist of an X-ray source, a sample holder, a detector, and a meter to control the angle of incidence. Data processing. After data collection, the raw diffraction pattern needs to be processed to extract meaningful information. The first step is to convert the recorded diffraction pattern into a two-dimensional image called a diffraction image. This image is then transferred into a one-dimensional diffraction pattern by integrating the intensity along a specific direction, usually the radial diffraction. Third, peak identification. The next step is to identify the peaks in the diffraction pattern. Each peak corresponds to a specific arrangement of atoms in the crystal. Lattice. The position, intestines, and shapes of the peaks provides information about the crystal structure, lattice parameter, and phase composition of the material. Peak identification can be done manually by comparing the observed diffraction pattern with the known patterns from a database or using automated peak fitting algorithms. Fourth one, data analysis. Once the peak are identified, various data analysis techniques can be applied to extract additional information. This may include determining the crystal structure using methods such as a rate-field refinement in calculating the crystal light size using the square equation, analyzing peak broadening to access strain or defects in the material, and quantifying phase composition using peak intensities or areas ratios. Fifth, interpretation and reporting. The final step is to interpret the results and report the findings. This involves comparing the observed diffraction pattern and derived parameters with known crystal structures or published data. The interpretation may involve a discussion of crystal symmetry, phase purity, latest distortions, and other relevant characteristics of the material. It's worth nothing that the data collection and analysis process in XRD can be complex, and there are various software packages available to assist with data processing and analysis. These software packages can often provide tools for peak fitting, data visualization, and advanced analysis technique. Now, fourth, force phase identification and crystal structure determination. Phase identification. After data analysis, the next goal is to identify the phases present in the sample. This is achieved by comparing the observed diffraction pattern with reference pattern from databases such as International Center of the Diffraction Data or the Cambridge Structural Database. The matching of peak position and intestines allow for the identification of known crystalline phases. Crystal structure determination. Once the phases are identified, the crystal structure of each phase needs to be determined. There are several methods for crystal structure determination depending on the complexity of the structure and the availability of additional information. The rate-field refinement methods is commonly used, which involves the refining the crystal structure parameters by minimizing the difference between the observed and calculated diffraction patterns. Other methods like direct methods are AB. Inertial structure determination may be employed in specific cases. Validation and refinement. After obtaining an initial crystal structure, it is important to validate and refine the results. This involves assessing the goodness of fit between the observed and calculated diffraction patterns, analyzing the agreement factors, and considering other indicators of data quality. Refinement techniques may be used to adjust the structural parameters further and improve the fit. Reporting the results. The final step is to report the phase identification and crystal structure determination findings. This typically involves documenting the identified phases, their crystal structures, lattice parameters, atomic positions, and any other relevant structural features. The results may be presented in the form of tables, diagrams, or crystallographic information files for dissemination to the scientific community. It is important to note that the process of phase identification and crystal structure determination can be complex and may require expertise in crystallography and XRD analysis. Specialized software packages like Foolproof GCS or Crystallize are often used to aid in the data analysis, peak fitting, and refinement steps. This is a diagram. Second, B single crystal XR diffraction. Single crystal X-ray diffraction is a powerful experimental technique used to determine the atomic structure of crystalline material. It provides detailed information about the arrangement of atoms within a crystal lattice, including bond length, bond angles, and the position of individual atoms. The process of single crystal X-ray diffraction involves several steps. First one, crystal preparation. A high quality single crystal of the material of interest needs to be obtained. They typically involves growing or selecting a suitable crystal with well defined faces and minimal defects. Second, mounting the crystal. The crystal is mounted on a geniometer which allows precise rotation and positioning of the crystal relative to the X-ray beam. Third, data collection. The crystal is exposed to a focused beam of X-rays which are rotated around the crystal at various angles. The diffracted X-rays are captured by a detector, usually a photograph film or a digital detector, producing a series of diffraction images. Fourth one, data analysis. The diffraction images are processed to determine the intestines and positions of the diffraction spots. The data are then used to calculate the structure factors, which are the amplitudes and phases of the diffracted waves. Fifth one, structure determination. The structure factors are used to combination with the computational methods to solve the crystal structure, typically by applying techniques such as direct methods. Patron methods are used to model building and refinement programs of sixth one, refinement and validation. The initial model of the crystal structure is refined to minimize the difference between the calculated and observed structure factors. The refined structure is then validated using various criteria including agreement factors, R factors, and goodness of fit parameters. This is a single crystal powder. See, thin film X-ray diffraction. Thin film X-ray diffraction is a technique used to analyze the crystallographic structure and orientation of thin films. Thin film are typically nanometers to micrometer thick layers of material deposited on a substrate, and TFXRD provides valuable information about their crystal structure, crystallography, texture, and lattice parameters. The basic principle of thin film X-ray diffraction is similar to single crystal X-ray diffraction, but the experimental setup and data analysis method are modified to accommodate the specific characteristics of thin film. This is a diagram of thin film X-ray diffraction. Here are the key aspects of thin film XRD. Experimental setup. Thin film samples are typically prepared by techniques like a physical vapor deposited, chemical vapor deposited, or sputtering. The thin film is deposited onto a substrate, which can be a single crystal water, the glass light, or another suitable material. The thin film and substrate together form a polycrystalline sample. Second, geometry. The X-ray beam is typically incident and at a grazing angle relative to a film surface. This grazing incidence geometry enhances the sensitivity to the film surface region and minimizes the contribution. Third one, diffraction and pattern analysis. The diffraction and the diffracted X-rays are collected and analyzed using a detector. The diffraction pattern is composed of a series of rings or ARC which corresponds to the diffraction from diffraction crystal planes in the thin film. By analyzing the position and intestines of the diffraction spots, information about the crystallography orientation, lattice spacing, and strain with the thin film can be obtained from the substrates. Texture analysis. Texture analysis Thin film often exhibit preferred crystallography orientation known as texture analysis involves determining the distribution and strength of crystallography orientation within the film. It can be achieved by measuring the intensity of diffraction peaks corresponding to specific crystallography planes and using specialized analysis techniques like pool figures or orientation distributed functions. Fifth one, data interpretation. The obtained diffraction data are typically compared with reference patterns from known crystal structures to identify the phases present in the thin film. The lattice parameters and crystallography orientation information can be extracted from the diffraction pattern using quantitative analysis methods. The thin film X-ray diffraction is widely used in material science, surface science, and thin film technology research. It provides valuable insight into the crystallographic properties of thin films, including their phase composition, crystal structure, crystallography texture, lattice strain, and defects. This information is critical for understanding and optimizing the functional properties of the thin film materials in various applications such as electronic, photovoltaic, coating, and sensors. Here's a diagram of thin film X-rays. How XRD parts are assembled. XRD or X-ray diffraction is a technique used to analyze the structure of materials by measuring the scattering of X-rays. The instruments used in XRD is called an X-ray diffractometers. It consists of several key components that work together to perform the analysis. Here is a general overview of how the parts are assembled in an XRD setup. One X-ray source. The X-ray diffractometer contains an X-ray tube that generates X-rays, typically a high energy X-ray source such as copper or cobalt anode is used. The X-ray tube is positioned in a way that directs the X-ray towards the sample. Second one, sample holder. The sample being analyzed is mounted on a sample holder. This holder can be rotated and tilted to achieve different orientation of the sample relative to X-ray beam. The sample holder is usually made of the material that does not significantly interfere with this X-ray scattering, such as quartz or glass. Third one, incident beam optics. To control the X-ray beam and focus it on the sample, the XRD setup includes incident beam optics. This typically consists of a series of colorimeters, slides, and filters that shape and filter the X-ray beam. The incident beam optics ensure that a well-defined and monochromatic X-ray beam tracks the samples. Fourth one, sample alignment stage. The XRD setup incorporates a sample alignment stage that allows precise positioning of the sample. The stage's adjustment in multiple directions to ensure accurate alignment of the sample with the incident of X-ray beam. With one detector, the scattered X-rays from the sample are detected using a detector. There are different types of detector used in XRD, such as scintillation detectors, proportional counters, or solid-state detectors. The detector captures the reflected X-rays and generates an electrical signal proportional to the intensity of the scattered X-rays. Sixth one, data acquisition system. The electrical signals from the detector are fed into a data acquisition system. This system converts the analog signals from the detector into a signal data that can be processed and analyzed by a computer and software. The XRD setup is connected to a computer that controls the instruments and performs data analysis. Specialized software is used to control the instruments parameters, collect the data, and analyze the X-ray diffraction patterns obtained from the sample. The software often includes tools for data processing, peak identification, and structure determination. It is important to note that the specific assembly of XRD components can vary depending on the instrument model and manufacturer. The above description provides a general overview of the key parts and their role in an XRD setup. How XRD instruments prepared. Preparing an XRD instruments involves several setups to ensure its proper functioning and accuracy. Here is a general outline of how XRD instruments are typically prepared. First one, instrument setup. Begin by setting up the XRD instruments in a stable and controlled environment. The instrument should be placed on a vibration-free table or platform to minimize external disturbances. Two, safety precautions. XRD instruments work with X-ray radiation, so it is critical to follow safety protocols. Ensure that appropriate safety measures are in place, such as radiation shielding, safety interlocks, and proper personal protective equipments for operators. Third, X-ray tube alignment. Proper alignment of the X-ray tube is essential for accurate measurements. Align the X-ray tube and adjust its position, focusing on the X-ray beam to ensure it is directed towards the sample holder. Fourth, sample preparation. Prepare the sample to be analyzed. The sample may need to be ground, polished, or prepared in a specific form depending on its nature. Example: when powder, thin film, single crystal. Follow appropriate sample preparation techniques and guidelines specific to the material under investigation. Fifth one, sample mounting. Mount the prepared sample on the sample holder depending on the instrument design. The sample may be affixed directly to the sample holder or placed in a specialized sample holder such as capillary or a thin film holder. Ensure that the sample is securely attached and properly aligned. Sixth one, incident beam optimization. Optimize the incident X-ray beam by adjusting the collimators, slides, slits, and filters in the incident beam path. This ensures that the X-ray beam is focused, monochromatics, and properly aligned with the sample. Seventh one, detector setup. Set up the X-ray detector in the proper position to capture the diffracted X-rays. Align the detector relative to the sample and optimize its parameters such as distance from the sample, detector angle, and detector resolution depending on the experimental requirements. Eighth one, calibration. Perform instruments calibration producers such as calibrating the two theta or other angular scale using standard references material with the known diffraction patterns. This calibration setup ensures accurate measurements and indexing of diffraction peaks. 9. Instrument validation. Before performing any sample measurements, it's essential to validate the instrument's performance. This can be done by measuring a well-characterized standard sample with the known diffraction patterns and comparing the results with established reference data. 10th, data execution setup. Set up the data execution system by configuring the parameters such as measurements, true time, setup size, scanning range, and detector setting. Ensure that the data execution software is properly connected to the instruments and ready to collect the X-ray diffraction data, and so on. XRD software features. XRD software features typically provides a range of tools and functionalities to process, analyze, and interpret XR diffraction data. Here are some common features found in XRD software. 1. Data import and conversion. XRD software allows the user to import raw XR diffraction data from various sources such as diffractometers and convert it into suitable format for analysis. Second, data processing and reduction. XRD software provides tools for processing and reducing the raw data to improve its quality and improve and remove artifacts. This may involve background substrates, subtraction, peak fitting, and data smoothing. Third one, data visualization. XRD software offers graphical tools to visualize X-ray diffraction patterns such as intensity plots and 2D or 3D representation. These visualization helps in identifying peak space identification and structure analysis. This is a diagram. Fourth one, peak identification and indexing. XRD software often includes databases of known crystal structures and allows user to compare their experimental data with these references for peak identification and indexing. This helps determine the crystal structure and composition of the material being studied. Fourth one, quantitative analysis. XRD software enables quantitative analysis of X-ray diffraction data such as determining phase abundances, crystallinity, and lattice parameters. It may include methods like refinement and profile fitting. Fifth one, structure analysis. XRD software provides tools for structure analysis, including determination of unit cell parameters, crystal symmetry, and atomic position. This can be done through techniques like full field analysis, partition analysis, and direct methods. Sixth one, texture analysis. Some XRD software packages offer functionality for texture analysis, which involves studying the peripheral orientation of crystalline grains in a material. Seventh one, reporting and export. XRD software allows user to generate reports and export the result of their analysis in various formats such as PDF, CSV, or Excel. It is important to note that there are various XRD software packages available, and each may have its own specific features and capabilities. Some popular commercial XRD software packages include Diffraction suite and so on. Now, application of XRD. There are different application of XRD. First one, material characteristics. XRD is widely used for the characterization of crystalline material. It can determine the crystal structure, lattice parameters, and phase composition of material, providing valuable information about the atomic arrangement and chemical composition. Second one, phase identification. XRD is employed to identify the different crystalline phases present in a material. The third one, quality control in materials. XRD is utilized in quality control process to ensure the consistency and integrity of materials. It can verify the process. this is a this is the main application of XRD. Fourth one, thin film analysis. XRD is used to study thin film and coating. It can determine the thickness, crystal structure, and preferred orientation of the deposited layers. XRD is particularly valuable in the semiconductor industry for characterizing the thin film structure used in electronic device. Fifth one is mineralogical analysis. XRD is employed in geology and mineralogy for the identification analysis of minerals. It helps to determine the minerals or composition of rocks and minerals. Physics one is pharmaceutical and drug analysis. XRD plays a critical role in the pharmaceutical industries for studying the crystal structure for drug compounds. It helps to understanding the physical properties of drugs such as stability, solubility, and which impact their formulation and efficiency. Forensic analysis. XRD is utilized the forensic science to identify unknown substances such as illicit drugs, explosive materials, and known unknown potters. Now, eighth one, archaeological and cultural heritage studies. XRD is applied to the analysis of archaeological, archivist, and cultural heritage materials. It helps in identifying the composition of ancient poetry, ceramics, pigments, and other materials. Ninth one, environmental analysis. XRD is used in environmental science to analyze the crystalline phases present in soil, sediments, and pollutants. Tenth one, nanomaterials and nanotechnology. XRD is critical in the characterization of nanomaterials, including nanoparticles, nanocomposites, and nanocrystalline materials. It provides information about the particle size, crystal structure, and lattice strain, which is essential for understanding and optimizing their properties and applications. There are just few examples of the diverse application of XRD. The technique is widely used across scientific research, material science, chemistry, physics, geology, pharmaceuticals, and numerous other fields. This is the diagram X-ray technique. Advanced and future directions of XRD. They are different. Here are some advanced and future direction of XRD. First one, high resolution XRD. Advancement in X-ray source detectors and optics have led to improved resolution in XRD measurements. High resolution XRD techniques allows for the analysis of extremely small crystalline features such as defects, strains, and interference in materials. In situ and operando studies. In situ and operando XRD technique involve analyzing material under real-time conditions such as during chemical reactions, phase transformation, or under applied external stimuli, that is, example given temperature, pressure, or electric field. Third one, time resolved XRD. Time resolved XRD techniques enables the investigation of ultra fast structural dynamics in materials on a picosecond to femtosecond time scale by combining XRD with the this is a diagram. Fourth one, grazing incident XRD. XRD is a technique used to study the thin films and surfaces by adjusting the incidence angle of the X-rays close to grazing incidence. The technique enhances the sensitivity to the near surface regions of materials. Fifth one, total scattering analysis. Total scattering analysis involves the combination of XRD data and pair distribution functions analysis technique. It allows for the study of both Bragg and diffuse scattering. Sixth one, and non-ambient XRD. Non-ambient XRD techniques involve studying material under controlled environmental conditions such as high or low temperatures, controlled humidity, or specific gas atmosphere. Seventh, micro and nano. nanoscale XRD. Advancement in XRD instrumentation have enabled the characterization of materials at micro and nanoscale dimensions. Micro XRD and nanoscale XRD technique offer the ability to analyze individual particles, nanocrystal, and small regions within a sample, providing insights into local crystallograph information, strain, and size effects. Eighth one, complementary techniques. XRD is often combined with other techniques such as ray, XRD, fluorescence spectrometry, XRA, X-ray photoelectron spectroscopy, and electron microscopy to obtain comprehensive information about the composition, chemical states, and morphology of materials. Ninth one, machine learning and data analysis. Advanced data analysis technique, including machine learning algorithms, are being employed to enhance data interpretation and analysis in XRD. Operando imagining. Combining XRD with imaging techniques such as XRA, commutative analogy allows for the three-dimensional mapping of crystalline phases and structural changes within materials. This is a diagram. These advanced and future directions in XRD are aimed at further enhancing its capabilities, enhancing the study of complex material and dynamic process at increasingly smaller scale and under realistic caption conditions. Continued development in X-ray source detectors, data analysis, and instrumentation will drive further advancement in the field of XRD. Now I define final conclusion of XRD. In conclusion, XR diffraction is a powerful and versatile technique used for the analysis and characterization of crystalline material. It provides valuable information about the crystal structure, phase composition, lattice parameters, and atomic arrangements within the materials. XRD has been widely employed in numerous scientific, industrial, and technological applications. The technique finds application in material science, chemistry, physics, biology, geology, and pharmaceutical, and many other fields. It plays a vital role in material characterization, phase identification, etc. Advancement in XRD instruments, techniques, and data analysis have opened new possibilities for the field. High resolution XRD in situ and open XRD time resolved XRD and combined techniques have provided deeper insight into material behavior, structural dynamics, and reaction mechanics. Furthermore, the integration of data analytics, mechanical machine learning and radiation sources and enhance the accuracy, efficiency, and capabilities of XRD. These advancement along the ongoing research in XRD methodology. Overall, XRD remains a fundamental and indispensable tool for material analysis, driving scientific advancements, technological innovations, and industrial developments. Its ability to reveal the structural properties of materials continues to contribute to our understanding of the world around us and supports advancement in various fields of research and industry. This is a diagram. No, thank you so much. That's all my sides.